my research interests, as has been described, is on the basis of trying to interface some of the more computational approaches alongside those that are more experimental to really make a meaningful set of connections between a disease state and the most effective cancer treatment. And the idea of cancer precision medicine paradigm is an attractive one. It's simple and elegant in that you are able to identify and match the appropriate treatment options for the relevant population. That's very wonderful goal to aspire for. However, we certainly now know that the idea of a genome-based precision guidance has been far from perfect, as faced a number of challenges, including that under even the best of circumstances, there are varied response, heterogeneities in response. Moreover, there have been pretty much consistently observed these acquired drug resistance, most likely as a function of tumor heterogeneity and cancer plasticity, which we still don't fully have a handle on. And perhaps really relevant then is the idea that many cancers lack meaningful actionable lesions. So what are we missing from all this? And so we have been thinking about this whole functional genomics really under the tip of the iceberg, really as a way to understand the actual consequence of these genomic changes and oncogenic events in the form of oncogenic pathways and cell states. And I won't get into too much of the details, but we have so I was fortunate to contribute to this review article concerning this functional universe of targets, and here's a little, I guess the cursor's not working, but here's a little diagram that converges a lot of these upstream events that apparently represents some of the work that I'll present today. So the idea is that oncogenes and genes and proteins in general don't operate in isolation, that they are always in a network of fellow genes and so on and so forth. So if we're able to capture and quantify those changes as a function of multiple genes, proteins, or et cetera, then we're able to much better recapitulate the activity of that oncogene. So that's really the basic concept, and we can take that even further and apply a more sophisticated way of capturing not only a single pathway, but using a fully data-driven approach to then rediscover or reconstruct the pathways that underlie a given set of tumors. Using this information and by cell states, really what we mean is the behaviors of multiple different pathways in a given sample. And once we're able to determine that, we have now proposed or devised a novel visual approach of being able to depict these samples relative to these pathway coordinates, almost like you would in a GPS with a quantitative value conferred with the proximity to each of these nodes that represent individual pathways, determining the location of the sample. So it's a fairly intuitive way to discern the cell state of a given sample. So here's one example, one that I described earlier, a classic example of BRAF mutant cancers being part of this, what we call the OncoGPS map. And you can already appreciate that there are three states that emerge from these BRAF mutant cancers, and note that the colors represent some of the classification of the groups with the corresponding pathways highlighted. And one thing that is of great use is then to determine whether or not these cancers will be sensitive to the agents that are targeting the BRAF variant cell. And so you can pretty clearly see that by implementing the genomic information with the cell state information, you can better predict sensitivity to the targeted therapy. Once you can determine these pathway activity, so I'm giving an example of an anti-oxidation pathway, the NRF2 pathway, for which once we can determine the quantitative measures of this pathway, we can also identify then in turn lesions that activate those pathways. So here's a study that we've completed where you can not only rediscover some of the lesions that are activating the known pathway activators as well as the negative regulators, but also find novel lesions that contribute to that ultimate activation of that pathway, serving as a powerful discovery tool to study the complex genome. So it's pretty clear to see then that integrating some of this information on top of the DNA information certainly has tremendous amount of value in making these kinds of predictions. And we would imagine that being able to integrate some of these sophisticated models will then allow us to make more precise tumor classifications, as well as better predict drug response, and as I'll get to, allow us to identify novel targets for therapy. And so more recently, we had an opportunity to apply some of these concepts. We were awarded this, we were one of the 3 institutes in California to be awarded this one-of-a-kind grant to allow us to try to conduct a pilot study in thinking about implementing precision cancer medicine. And I won't have, there are a lot of moving parts. There's the community outreach and there's the, clinical interactions and so on and so forth. I won't have time to get into too much of the details, but certainly worth mentioning that, one of the meaningful aspects of having executed this is to be able to think of a lot of the things that we do in the context of, real translation, not simply for purposes of academic research, but thinking about how these studies, these preclinical studies, impact patient care was something that we care deeply about. And I believe the lessons that we learned through this process could apply to many other diseases. Now, we focused on, for this project, breast cancer, not because we're breast cancer researchers per se, but really as a benchmark disease for which there have been some very well established clinical and molecular guidelines for treatments. And there have been some, again, clinical assays as well as genomic based assays. Certainly, in some of the subtypes, we find that there are a tremendous amount of molecular heterogeneity as it comes to genomic changes, and certainly has not been a reliable way for classifying these diseases. So without getting into too much of the details, when we look at breast cancer as a whole, you find that there are 3 states that emerge quite prominently, and they seem to align rather well with the conventional wisdoms, except a number of key things really popped out at us. For the interest of time, I'm giving one example in which we find that if you look at the HER2 cancers, which really define a category of its own, given the availability of some of the therapeutics, that they actually diverge into 2 of these cell states. And when we look at these diverging cell states, we would predict that they would have some different functional consequences. And so one of the ways to test this, and in fact is a very powerful aspects of this approach, is then being able to bring in external data sets into this model. And so here you're looking at a study that was a multi-centered clinical trial-based study that have produced a number of this data, and certainly HER2 positive patients were recruited as one arm of the study, were given the standard of care HER2 inhibition as well as chemotherapy combination. And so we were able to integrate that data and ask the question, well, do these separate states really tell us something about the response, at least to the standard of care? So when we project that data onto our model, what we find is that as opposed to roughly 26% response rate as determined by the pathological complete response, when we look at these groups, we find that this group right here significantly increases the pathologically complete response rate, which is quite reassuring given the fact that they are, again, functionally very different. Now, we went further, and much like you would in a map setting where you would then zoom in, we were also further able to zoom into this group to get a more higher resolution differences among these breast cancers, and found that these quote unquote these triple negative breast cancer states were then further divided into 4 groups. Now, if you apply that very same data and further classify on the basis of these classifications, you do find then that there are yet another 14% increase in this response. And collectively, whereas the unselected population, you have about 26% response. When you put this into this model, you're able to get almost 2-fold increase in being able to predict response. And certainly it's not perfect, but I would say it's a significant improvement. Now back to this triple negative breast cancer setup, where if you actually look, triple negative breast cancers, as we know, tend to have a poor prognosis compared to other groups. But if you now zoom into some of these subgroups, you find that there's additional differences in the prognosis. And these 2 that I've highlighted happens to have the worst prognosis. And we've done a considerable amount of characterization in these groups. But the most notable thing that emerged from some of the analysis was the fact that one of these poor prognosis group actually was significantly enriched with an African American patient population, whereas the other group has more significant enrichment in European, those of European ancestry. And I do highlight that this is an enrichment and not a all or none, as we have continued to see in a lot of sample-based assessment. And certainly it's a meaningful one because there has been a great deal of interest in understanding the sources of disparity in some of the African American triple negative breast cancer patients. And our model appears to be unique in being able to identify this very meaningful associations, whereas others have not. We also found that San Diego is a heavy Hispanic community, and the patients from Hispanic ancestry are significantly underrepresented despite tremendous amount of data sets available. And so we have collaborated with some of our community hospitals to sort out some of the infrastructures and logistics to collect a number of samples. And across several different data sets, including our own, we find that Hispanic patients also are enriched in this poor prognosis category. And certainly work of Laura Fireman and others have shown that there are higher incidence of triple negative breast cancer in Hispanic patients. So this all gives us now a potential handle into the underlying biology. Of course, one could argue there's a lot of complex confounding factors that play into this, but certainly there appears to be some biological underpinning that could be further explored. One interesting set of associations that we find are the enrichment for UV signature and mixed signature in these groups. Notably, these 2 groups appear to be very different in their immune presence. and something that we're very interested in certainly following up, and I'll touch upon this a little later. This is going to be something that we're going to look into more closely at the level of the genome in collaboration with Elad Ziv at UCSF. So yet we can further zoom in. This is something that's recent development where we can not only zoom in into those bulk territory, we can look at each of the single cells, map those single cells as a bulk into these maps, and really get an understanding of what those states represent in terms of the cellular constituents. And here's one example as to why that would be meaningful. And so here's again that example of HER2 cancers. When you zoom in, not only do you find that there are differences in the immune and the stromal constituents, but you also find both qualitative and quantitative differences in the cancer cells and their corresponding states. So that confirms this, that initial description of this divergent state of this oncogenic driven tumors. Moreover, this is really quite exciting to be able to then simply hone in on the cancer cells and make meaningful associations with data that we have established for cancer cell lines. And, as intuitive as it might sound, there have been a lot of preclinical studies performed on cell lines that have not well translated in the clinic. And, one of the point of contention was that perhaps cell lines are not good models for human cancers, when in fact that's it's really comparing apples to oranges. And so in this setting we can actually take those cancer cells and we can do a more apples to apples comparisons. And the reason why that's meaningful then is that there are now a tremendous amount of molecular data that's been assembled, including drug response and genetic vulnerability assessments. This CRISPR screen has been now increased up to 1,000 cell lines. And so we can really harness the power of the functional genomics performed across these cell lines to then come up with an incredibly exciting and novel therapeutic hypothesis. And I won't have too much time to go into details. This is something that we've spent a considerable amount of time, and even with cell lines in the laboratory, there's an enormous amount of complexities as you really explore the cell circuits and whatnot. And we have taken some of these agents and performed a number of preclinical assays, including cell lines from breast, as well as patient-derived organoid models that we've collected in collaboration at UCSD and Moore's Cancer Center physicians, as well as the biorepository. And just to give an example, here's one so-called in silico screen that allowed us to identify FGFR as one of the top targets hitting one of these major pathways. And it's quite reassuring then that out of 20,000 genes, different orthogonal genetic screens have identified as top hits the very same gene, as well as the drug. So it gives us a considerable amount of confidence. And lo and behold, when we go and test some of these out in the lab, that they are quite effective. And I do want to point out that despite these predictions holding up, one thing that we have come to fully appreciate is that this is probably good enough for publication, but not so much if you're thinking about, well, how do we think about translating these findings? Because a lot of times, and in fact, more the rule than the exception, there are a lot of residual disease. There's not been a single agent, even in a cell in a dish, that completely wipes out these cancer cells. And one of the interest in the field has been this persister state, these cancer cells that cling on to then go on to form a much more aggressive disease, drug resistant, and perhaps even from a more or less a dormant state into metastasis. So something that we've been very interested in for following up. And certainly a lot of clinical trials, even the ones that are being conducted more recently, have iteratively found that single agents, especially in the more aggressive disease settings, are not as effective. And so one of the nice things that we were able to deploy using this kind of approach then is to nominate potential drug combinations. And indeed, in this setting, we're able to connect them to pathways, make predictions about agents, and to combine them in a way that displays a considerable amount of synergy that's sufficient to kill a lot of the residual cells. Now, I just want to highlight some of the recent work by UK Sanger Institute have performed a very brute force drug combination screen. And one of the take homes from that, other than that, it's a huge scale, extensive experiment across drugs, is the fact that synergies are observed significantly less than expected by chance. So certainly, if you want to find these synergies, you'd be doing a lot of these studies across many different systems, only to find those handful of synergies that emerge. So having these kinds of machine to guide that process has certainly been incredibly valuable. And I know that you're mostly not as interested in breast, but I would argue that a lot of the lessons and approaches, as well as some of the biologies, actually are well applicable across many other cancers. And this is something that we've found to be the case as we look at sort of the pan-cancer landscape, that there are certain pathways that are quite preserved. And of course, there are very, very specific disease nuances, which experts like yourselves contribute to the enormous amount of valuable insights. But I do want to highlight that this early examination of or assessment of prostate cancer was quite interesting, and I do want to highlight it's a preliminary, but applying the similar set of approaches, we found that in prostate cancer there appears to be at the top level 3 groups that emerge, and these groups appear to be preserved also in the settings of different prostate cancers that were collected across the continent. And one of the things that that emerged was that these prostate cancer groups appear to have differences in their survival characteristics. It's much more reassuring to find that characteristics is not only preserved but are highlighted in other data sets, which is always reassuring. One of the really interesting things that emerges from these kinds of analysis is the ability to look at some of the pathways and its tumor microenvironment settings. And one of the things that we found in this worst prognosis group is, in fact, that there's a significant increase in leukocyte infiltration score, as well as the STAT1 pathway activation. And I'm sure that many of you are familiar with work by Chuck Sawyers, where this JAK-STAT's inflammatory signaling appears to be one of the underlying causes for cell state plasticity. Now, if you look at this other group that has just as bad of a prognosis, you find that it's quite the opposite, that they're rather immune cold. If you look at both of the leukocytes as well as the pathways. But one striking thing that emerges is then when you apply this pan-cancer derived stemness signature, it is highly enriched in the green group as compared to the others. Again, this is a pan-cancer study that applied machine learning to identify the most salient aspects of stemness features and found this to be the case. And equally interesting is something that we have been looking at for quite some time, this idea of metastasis states. So here's a study by Arul Shanayan at the U of Michigan, who's done a Herculean task of collecting 500 or more metastases from patients across 20 or so different tissue types, both of primary and of the sites. These are metastatic tumors. And one of the really striking things that emerged as we deconvolute some of the pathways that emerge from this analysis is the fact that prostate cancer appears to be actually one of the two diseases that are incredibly homogeneous with respect to its program. The second one is the breast cancer, which appears to be equally, but perhaps a little less homogeneous. And so the question is, is there some kind of an intrinsic metastatic program that we could relate back to the primary tumors? Again, these are signals from metastasis, and when you go back and apply this, you find at the bottom panel here that this stemness group also appears to be significantly enriched for metastasis signature. So I think that's very interesting. Obviously, the same set of tools and approaches apply where we're now able to hone in on some of these states in a more defined resolution, including at the level of single cells, and to parse out some of the immune cells, as well as being able to perform some of the primary metastasis matched analysis, which provides more of a direct apples to apples comparison, and certainly gives us now the basis for then nominating some additional candidate targets to pursue. Now, this I wouldn't share. It's quite underpowered, as you can see on the right-hand panel here. If you look at the ethnic representation, and the black dots represent those from African ancestry, I would not show it as significantly underpowered had it not for the breast cancer result and some parallels to the immune sort of enrichment, as well as some of the biological underpinnings. And so, we're quite, again, interested in this idea of, is there some biological basis for some of these disparities? And it would be something of an opportunity to be able to, ask that question in a more direct way. Again, we're very interested in this, this ability to target in synergies. Certainly, we have seen that cancer cells exist in multiple clonalities, and as you can see, that's actually real data, where you see two distinct populations of cancer cells within a tumor, and the idea would be, well, do we need to combine 4 drugs? And it's something that's quite unfeasible and not ideal. So, as with many that are interested in this idea of harnessing the immune system, we have been collaborating with a number of people trying to harness both the CD8 T cells as well as NK cells. Notably, some of these studies really are going after the cancer intrinsic aspects that, if you target a certain pathway, can you change the way that they would respond to some of these cytotoxic immune cells? And more recently, on the right panel here, we find that quite remarkably, the presentation genes, the antigen presentation genes, are highly coordinately regulated, which speaks to a more or less a coordinated program. And we found recently that we can actually, in some of these underrepresented cells, the cells that express very, very low levels of MHCs, can be reverted through some of these drugs that we have identified through some of these analyses. So something that it would be of great interest to further follow up in a more relevant setting. And to that effect, I've spent a lot of time on, discussing how we can break cancers. But one of my key interests is also being able to make cancers from scratch, because we believe being able to make cancers might allow us to get insights into how we can break them. And so here's some of the studies that we've performed over the years, really taking this landmark study from Dr. Bob Weinberg and in being able to transform normal epithelial cells into cancer. And we've done quite a systematic characterization, both at the level of some of these cell state and pathway changes, as well as looking at phosphoproteomic changes. And it's an incredibly valuable resource to be able to not only study, some of the potential targeting opportunities, but also to study cancer initiation and maintenance. The only caveat, as we can imagine, is them being used in the absence of the relevant immune system. So here, this is a work in progress. The idea is, can we leverage, and the other bottleneck, by the way, is how do you make these cancer cells? What are the so-called ingredients? And we have found that it really takes again, set of synergistic combinations to achieve this full oncogenic transformation potential. So the idea is, can we harness the incredible amount of data that's available with respect to cancer? You apply the machine learning approaches, some of which I've described, to then nominate candidate events, which would, in synergy, allow a normal cell to really undergo cellular transformation. And we're doing this in the context of being able to deploy this in an immunocompetent setting, to be able to study not only obviously the cancer initiation, but how that then affects the immune and vice versa. And on the right is a preliminary analysis where we were able to hone in now on a specific set of cancer cells from that analysis that have some of these hallmark lesions in the form of copy number changes that then can be hopefully mimicked in a mammary organoid setting that we're currently culturing and transfecting for these changes to occur. Like I mentioned, there have been a lot of discussions as to how to model systems best to recapitulate cancer, but certainly our models have been significantly superior in being able to parse out some of the nuance differences, and something that we are able to then bring in some of the tumors from mice, for instance, and connect them directly to human equivalent diseases using this kind of approach. So we're excited to continue to apply this kind of approach for development of both experimental as well as computational models for future efforts. So with that, I want to end with this idea that, you know, if nothing else, what we've learned is cancer synergize at all levels, whether it's at the cellular level or at the level of the pathways. And they're extremely resilient. And one thing that, you know, we came in the lab, we were frustrated, and then we were, you know, we're declaring victory one day because now, you know, it works, and then the next day we're wrong. And, you know, it's an incredibly complex system. And I think that the way that we can, you know, tackle this problem is really reconnecting, you know, coming together as a community for a very, very effective collaborative model, and certainly deserves redefining a lot of what we think are the defaults. You know, here are the classification schemes that happened a while ago, and so are we not going to question? And my experience has been such that we need to revisit and redefine a lot of things that we think we know for a fact, because within those are the real essence of discovery. And I want to end by this map of Columbus that was used to navigate and discover the New World. And, you know, it's sort of where do we see ourselves in the grand scheme of things? And I'd say probably we have one of these maps. It's not by all means perfect. We're missing a lot of things, but it certainly allowed us to make the significant discovery. By the way, this past Monday was Columbus and Indigenous Day, so it's quite relevant. But I think that that's the spirit that we can use some of these maps to allow us to then really make these significant discoveries. So obviously there are a lot of people to thank. This has been an incredible team effort. Some of the people in my lab have done a lot of the work. During the COVID, we had a lot of difficulties, and I'm sure everybody did, but they stuck to it and really pursued a lot of these programs. And I want to thank some of the collaborators. This is actually pre-COVID, where we were invited to the governor's office to as an inaugural meeting, and we didn't expect that to be kind of the last, because going in, we didn't expect COVID to happen. But anyway, thank you so much, and we'll take any questions that you might have.
It seems to me that that as we get better and better at precision medicine and substratifying cancers by phenotype or these molecular subclassifications, the really common cancers like bladder and prostate and breast are going to begin to face some of the problems that folks who study rare cancers face, which is it's very hard to do clinical trials in cancers or groups that are smaller and smaller and rarer and rarer. I think we've seen this be a challenge in clinically translating some of the findings in bladder cancer and so on. So I guess my question is, when you look down the road 10, 15 years, with that challenge, how do you bring some of these challenges into the clinic when you can't run randomized trials on subgroups and subgroups and subgroups?
That's an excellent question, and something that we thought about considerably because, a lot of the settings that you described, it's really kind of a more focused approach to, these trials. We had initially envisioned a more or less an umbrella type of trial where, it's really on a sample by sample, basis. Obviously, it has multiple challenges and did not end up that way. But one of the really powerful things about the functional genomic approach is that you're essentially using those pathway information as currency to other diseases. So if you find evidence of some of the common pathways altered in these rarer diseases, which we do, and we certainly have looked at some of these rare, some of the pediatric diseases, and et cetera, they, in the end, cancer only can dysregulate so many pathways and genes and so on and so forth. So we've had some, degree of success in, picking up those signals. Now, I think really the value in a lot of what we do has been the exceptions, and it's been rare that we don't, in fact, I don't think we've ever seen a case where there hasn't been an exception in cancer research, whether it's cell lines or, et cetera. And those exceptions always prove to be extremely valuable. And so, one of the paradigms really was conceived as a function of being able to use that exception to gain new knowledge. And so I think that, that's another way I think could be approached. The third important thing that I think I want to sort of highlight is the fact that some of these rare diseases or some of the more common diseases, we learn considerable amount of biology by perturbing them. So a lot of what I've shown are these steady state diseases that are profiled as such, but when you actually perform some kind of a drug treatment and find these changes that occur, we've actually gained a lot more by that one axis of a dynamic change than across 100 samples. And so we certainly think that could be another opportunity, but certainly, a lot of challenges moving forward. I mean, this is really kind of a glass half empty situation in my view, which is to say we have a lot of exciting work to do, but hopefully that answered your question.
Yeah. I have a couple questions. And first of all, using the functional genomics and use that to tease out the clinical response to patients. So ultimately, at the end, it's going to be whether that can be utilized clinically. So what effort is it going on right now at San Diego to utilize this approach to help manage our patients?
We were one of the 12 centers across the country to come together with this idea of, functional genomic-based precision medicine. And I can tell you across the board, there remains certain level of infrastructural perhaps or administrative challenges to integrate some of those concepts. And again, I mean, this is happening across many institutes. I do think there's a lot of opportunities, but the idea is, what will it take for us to really kind of change the course of thinking about this problem well beyond the genome-guided path? And a lot of institutes still only collect genomic information at the level of DNA. So often, Tempest, which is a big, player in this, would mostly collect DNA information, and that by itself, we think, is less power to make these kinds of more fruitful decisions. But certainly something that appears to be less of a scientific, more of a a sort of a challenge moving forward to be able to deploy these kinds of thinking.
No, then thank you for that response. The second thing question is on the organoids, because we were looking for some sort of connection between the genomic data that we have, and as they see a bridge to the clinic using the organoid model to validate or test, right? To that end, when you have such a heterogeneity in what you're seeing clinically, how does organoids and models like that would work, and how can you develop, what kind of models are out there that better predicts or more useful, I guess?
Yeah, it's very interesting that you bring that up, and it actually pertains to that earlier question about rare diseases. And being part of this network allows us to look at a lot of different spectrum of assays towards the goal of precision medicine implementation. And one of the groups have certainly used organoids in a rare disease setting, a target that was absolutely novel and appear to have some degree of success. So I think it really comes down to, and I know work by Calvin Kuo and at Stanford have sort of pushed the envelope in being able to preserve the immune system in certain settings, although that's quite limited to a certain number of passages. So I honestly think that it really comes down to how well does the disease recapitulate the actual disease. And certainly, a lot of people are now invested in asking the same questions in PDXs. How well does the PDX recapitulate the human disease? Our approach is that all models are wrong. We have to know exactly what is being modeled to then be able to really translate some of those findings in the clinic. And that's something that we've come to realize is the more important and perhaps a faster way to connect and make those connections meaningful than to somehow believe that there is a perfect model. So the idea would be, can we use multiple orthogonal models to convince ourselves that the treatment strategy is indeed suppressing the relevant pathway, changing the functions of the relevant biomarkers to then be able to connect the dots in a more sort of clinical setting. So that's kind of how we're thinking about this problem, more so that, and I do want to just sort of end with, to your question, we did look across a lot of organoids in pancreatic cancer at one point. And I'm sure there's a lot of work put in to try to simply establish an organoid, because the irony of all this is that they die. And so we want them alive so we can kill them. But a lot of the ingredients that we use apparently are biased towards certain cell types and not others. And so what we ended up seeing was, and this was a work in collaboration with Hervé Thiriac and Andy Lowy, that across 80 panel of pancreatic cancer organoids that were established, there was an underrepresentation of certain type of pancreatic cancer as opposed to others. And so there is clearly some kind of a bias, even through culturing and collecting these biospecimens. So it's something that I think we need to also pay attention. And all the more highlights the fact that we need to look at functional equivalent changes that happen in all these models. Thank you.